Okay, so we talked about excitation-contraction coupling last time. Now we're going to talk about cross-bridge cycling. So here's the topic and outcomes. Okay, the sliding filament theory is what explains how cross-bridge cycling occurs. Okay, and then we're going to talk a little bit about skeletal muscle contraction. So to briefly summarize what we talked about last time, in order for a muscle to contract, the following things need to occur. We need an action potential to come down the neuron. This will ultimately release acetylcholine, which activates ligand-gated sodium channels and allows sodium to flow into the muscle cell. This causes a graded potential, which opens up voltage-gated sodium channels and allows even more sodium influx, which allows threshold to be hit, which causes an action potential to occur. This action potential travels along the sarcolemma, or cell membrane of the muscle cell, and then down into the T-tubules. When the action potential goes down into the T-tubules, it activates the DHP receptor, which opens a voltage-gated calcium channel, and this allows calcium influx. The activation of that DHP receptor also activates a ryanodine channel, and that ryanodine channel is on the sarcoplasmic reticulum, which allows calcium to leave the sarcoplasmic reticulum and go into the sarcoplasm. Calcium then binds to troponin. This moves tropomyosin out of the way so that the binding sites on actin are open and myosin binds to actin. This process is called excitation-contraction coupling because there's a link between the electrical stimulus or excitation and the mechanical movement of the muscle cell or the contraction. Okay. This binding or this formation of cross bridges leads to tension in the muscle, but we're not contracting the muscle at this time. So as we discussed previously, a sarcomere shortens when the myosin heads, shown in purple, pull the actin fibers, shown in green, towards the M line. This process is explained by cross-bridge cycling. Remember, calcium entered the sarcoplasm, which allows actin and myosin to bind together tightly. When a myosin head is bind to an actin fiber, we call this a cross bridge. In order for a muscle to contract, myosin is going to have to pull actin, detach from actin, reattach to actin, pull actin, and repeat this process over and over. In order for this to happen, we need ATP. As we go through this process of cross-bridge cycling, I want you to remember that ATP and its products are going to cause something to occur with myosin and actin. So, in other words, ATP is the cause, myosin is the effect. So at this point, actin and myosin are bound together tightly. So as you can see from this diagram, an ATP molecule binds to the myosin head, and that causes actin and myosin to break apart. And I know some of you are thinking, wait a minute, they were just bound together. Absolutely. But we're going to get to that. So the cause, ATP binds to myosin. The effect is actin and myosin separate. Once bound to the myosin head, ATP is broken down, which releases energy. Okay, and this breakdown occurs with the help of an enzyme called ATPase. It's actually myosin ATPase. Okay, the release of this energy causes the myosin head to cock or change its position, and ultimately this allows actin and myosin to reattach. Cause, energy is released from ATP. Effect, myosin head cocks and actin and myosin reattach. Next, we release phosphate's released, which causes the power stroke or pulling of the actin towards the M line. When phosphate's released, the myosin head essentially becomes uncocked, and as it becomes uncocked, it pulls that actin towards the M line. Finally, ADP is released, which results in the tight binding of actin and myosin. And this process will continue. These 4 steps will continue, and the muscle will shorten as long as we have 3 things present: acetylcholine, calcium, and ATP. If any of those 3 things is missing, it's going to be difficult to maintain muscle contraction or maintain cross-bridge cycling. Acetylcholine is released by the neuron, which causes an action potential in the muscle cell. This leads to the release of calcium, which allows actin and myosin to bind together. And then we have ATP, which is needed for that cross-bridge cycling to occur. Okay. When an action potential stops, this allows muscle cells to repolarize, the ryanodine receptor closes, and calcium goes back into the sarcoplasm via the calcium ATPase pump, And as a result, actin and myosin, it's more difficult for them to bind. And so we reduce muscle tension. There's still calcium and there's still some cross bridges, but it's to a much less or much smaller degree. This is what leads to this reduced muscle tension. So, can you do the following two things? Can you summarize how cross-bridge cycling occurs? And again, I would challenge you to see if you can draw something out as opposed to just listing it step by step. And can you describe how muscle relaxation occurs?